Today, we are going to start with the aromatic amino acid metabolism part 2. In a previous lecture, we have gone through the metabolism of phenylalanine and tyrosine. Today, we will learn about metabolism of tryptophan. Tryptophan is an heterocyclic aromatic amino acid. You can see there is a heterocyclic structure of tryptophan. It contains indole ring. This is very important, you have to remember. Sometimes it asks in a MCQ which group or which ring the tryptophan amino acid contains, that is indole ring. It is an essential amino acid and metabolically it's both ketogenic as well as glucogenic. Tryptophan is a very important amino acid because there are a list of biologically important compound that is synthesized from tryptophan as a part of metabolism of tryptophan. We'll see here is a list of that important compounds. One is alanine, that is derived from tryptophan metabolism, and we know alanine goes to the gluconeogenesis, hence it is a glucogenic amino acid. Acetoacetyl-CoA that splits to acetyl-CoA and it forms ketone bodies, so it's a ketogenic amino acid. Other products, it produces the formyl group that helps in the 1-carbon metabolism. It produces the niacin, that is a B complex vitamin water soluble compound, and from the niacin we'll get NAD+, that is a coenzyme form of niacin. Serotonin and melatonin, that are the neurotransmitters synthesized from tryptophan. Then hydroxyindole acetic acid and indicant, they are the end product of tryptophan. Now we'll see the tryptophan metabolism and how the products synthesize from the tryptophan one by one. The first step, tryptophan metabolized by tryptophan pyrrolase to N-formyl kynurenine in the presence of oxygen. Then this N-formyl kynurenine converted into kynurenine with the help of kynurenine formidase or formylase enzyme. Here in this step we can see there is a release of 1-carbon compound that is format, that gives to the 1-carbon metabolism or 1-carbon pool. So this is the first compound that we get from the tryptophan. In the next step, this kynurenine get converted into 3-hydroxykynurenine with the help of kynurenine hydroxylase enzyme, and it requires NADPH as a coenzyme. Then this 3-hydroxykynurenine gets converted into 3-hydroxyanthranilic and splits an alanine with the help of kynureninase enzyme. This is very important step of tryptophan metabolism. And for this reaction, what we required is B6. So in this step, what we get is 3-hydroxyanthranilic acid and the alanine. This alanine goes to the gluconeogenesis. By transamination, it gets converted into pyruvate, and pyruvate goes to the glucose synthesis. So this is the another product that derived from the tryptophan. This 3-hydroxykynurenine and the kynurenine also get converted into xanthurenic acid with the help of amino transferase group of enzyme. And this xanthurenate or xanthurenic acid excreted in the urine. Now, this 3-hydroxyanthranilin takes an alternate pathway and it forms a niacin, and this quinolinic acid is an intermediate of this pathway. That is one way of metabolism of 3-hydroxyanthranilic acid. This niacin get converted into, depending upon requirement, NAD plus and NADP plus. The other way of the metabolism of 3-hydroxyanthranilic acid is it goes to the converted into amino cis-muconate semialdehyde, then ultimately to the oxalocrotonate, alpha ketoadipate, glutaryl CoA, and then to the acetoacetyl CoA. This is ketogenic and that get converted into acetyl CoA. So by the metabolism of tryptophan, first we will get one carbon compound, that is formaldehyde, or the format, that is one carbon compound. Then we get alanine, that is a glucogenic compound. Then we get niacin, that is a water soluble vitamin, and then acetyl CoA, that is a ketogenic role. As we had seen previous slide, this 3-hydroxyanthranilic acid take an alternate route and metabolize to quinolinic acid and to, by the spontaneous reaction, converted to niacin or nicotinic acid. Then this nicotinic acid react with the PRPP phosphoribosyl pyrophosphate in the presence of quinolinate phosphoribosyl transferase, that is QPRTS. It converted into nicotinamide mononucleotide. Then it converted into desamido NAD plus with the help of ATP, and again with the help of glutamine it gets converted into NAD plus. So this is the coenzyme form that we get from the niacin, and this niacin we get from the tryptophan. Now about 3% molecule of total tryptophan, whatever we are taking, they will diverted at the level of 3-hydroxyanthranilic acid to form this NAD plus. About 60 mg of tryptophan what we require to produce 1 mg of nicotinic acid, and for that vitamin B6 is important. In previous reaction we have seen that from the kynurenine, when this hydroxyanthranilic acid form, B6 is required. Hence even in the B6 deficiency we will not get this 3-hydroxyanthranilic acid, so we will not able to produce niacin, and this resulting in niacin deficiency. Now the other products we get from the tryptophan, that is neurotransmitter, melatonin and serotonin. The tryptophan, with the help of tryptophan hydroxylase, like that of phenylalanine hydroxylase in phenylalanine metabolism, this tryptophan with the help of tryptophan hydroxylase get converted into 5-hydroxytryptophan, and it also required tetrahydrobiopterin. In the next step, this 5-hydroxytryptophan decarboxylated in the presence of pyridoxal phosphate to 5-hydroxytryptamine, that is also known as serotonin. This serotonin degraded by monoamino oxidase, that is MAO enzyme, and it forms 5-hydroxyindole acetic acid. This is an end product of tryptophan metabolism when it goes to the serotonin synthesis. Then serotonin acetylated with the help of acetylase enzyme. It forms acetyl serotonin. In the next step, there is a methyl transfer from the S-adenosyl methionine to the acetyl serotonin, and here you can see it forms a melatonin. So in the presence of S-adenosyl methionine, this acetyl serotonin converted into melatonin. So these are the two neurotransmitters, serotonin and melatonin, synthesized from the tryptophan. Now the serotonin, that is also known as 5-hydroxytryptamine, It is a very important neurotransmitter in brain and it has an antidepressant property. It's a vasoconstrictor substance and synthesized mainly from gastric mucosa, intestine, brain, mast cells and platelets. The serotonin is involved mainly in mood, sleep, appetite and temperature regulation. It increases the GI motility. It is also reduces the pain sensitivity. Now, the applied aspect associated with the tryptophan metabolism, that is carcinoids. It is also known as a argentaphenomas. What is that? It is a malignant tumor of serotonin producing cells. The clinical feature associated with this tumor, that is carcinoid, is known as carcinoid syndrome. In carcinoid syndrome, up to 60% of tryptophan is diverted to serotonin production. So most of the tryptophan we have that will divert it to the serotonin production. And because of this excess serotonin, it leads to symptoms like flushing, sweating, intermittent diarrhea, respiratory distress, bronchospasm, and the fluctuating hypertension. This can be diagnosed by estimating the urinary excretion of 5-hydroxy-indole acetic acid. We have seen that the serotonin get converted into 5-hydroxy-indole acetic acid with the help of MAO, that is monoamino oxidase, and that is excreted in urine. Normally it is 2 to 7 milligram per day, but in case of carcinoid syndrome, its excretion increase as much as 400 milligram per day. So it is a very important diagnostic test for the argentaphenomas or the carcinoids. Now let's see the relationship of tryptophan and sleep. It is in terms of constituents of diet. For example, we take carbohydrate rich diet, it induces the sleep. As we know, if we take much of rice and the other carbohydrates in diet, it induces the sleep. While the protein rich diet is associated with the alertness. Let's see the reason behind that. When we take protein rich diet, at the time there are so many amino acids in the blood. And these amino acids competes with the tryptophan for transport across the blood-brain barrier, resulting in lowering the availability of tryptophan for the serotonin production in the brain. And we know the serotonin that induces the sleep. So in that condition, when we take protein-rich diet, because of this competition between the amino acid, less tryptophan enters into the brain, less serotonin is produced, and that does not induce the sleep. While when we take carbohydrate-rich diet, it releases the insulin. That insulin causes uptake of amino acids from blood to the skeletal muscle, resulting in decreased competition between the amino acid to enter in the blood-brain barrier or to cross the blood-brain barrier. So the tryptophan easily enters into the brain. It produces the serotonin, and the serotonin induces the sleep. Because of that, carbohydrate-rich diet induces the sleep. Melatonin, that is the second neurotransmitter we have seen synthesized from the tryptophan. It is the hormone produced by the pineal gland and synthesized from the serotonin by the methylation reaction. The functions of melatonin, it is synthesized at night and is involved in circadian rhythm. So it regulates the process of sleep and wakefulness. So when to sleep, when to wake up, when we get hungry, it's all the rhythm that what is controlled by melatonin. This melatonin inhibits the secretion of MSH, that is melanocyte stimulating hormone, and ACTH. This melatonin also acts as a neurotransmitter by inhibiting the synthesis of other neurotransmitters like GABA and dopamine. So these are the functions of melatonin. Hartnup disease. That is an autosomal recessive condition associated with the tryptophan metabolism. Particularly, it is associated with the defective absorption and reabsorption of aromatic amino acid, mainly the tryptophan from intestine and renal tubule. So there is an increased excretion of tryptophan. There is a decreased availability of tryptophan, so there is a decreased synthesis of niacin. We know niacin synthesis or the niacin deficiency leads to pellagra. So the patient develops pellagra-like symptoms due to impaired synthesis of niacin from tryptophan. Person also suffers from dermatitis, ataxia, abnormal behavior, and this tryptophan and the other aromatic amino acid excreted in urine. Patient diagnosed mostly based on amino aciduria. We can detect the amino acid excretion in the urine by the chromatography technique. And the increased excretion of this indole compound, that is also detected by Obermeyer test, that is a diagnostic test for the Hartnup disease. So this is about the summary of tryptophan metabolism. How we getting different products from the tryptophan metabolism. First we get is a formal tetrahydrofolic acid, that is a 1-carbonamide compound. We get serotonin and melatonin. There's a neurotransmitter. This 5-hydroxyindole acetic acid and the indican, that is an end product or the degradation product of tryptophan. As we know, tryptophan is glucogenic and ketogenic because it produces the alanine that produces the glucose by the gluconeogenesis pathway, and it produces the acetoacetate that goes to the ketogenic pathway. 3-hydroxyanthranilic acid also produces the niacin, and it produces the coenzyme of niacin, that is NAD+. So this is all about the tryptophan metabolism. Thank you so much. So we have concluded the metabolism of aromatic amino acids. In the previous class, we have learned about phenylalanine and tyrosine, and in this class, we have learned tryptophan metabolism. Okay.